This report examines the history, production, and the step-by-step process of creating GMOs. Understanding GMOs development is essential for evaluating their impact on agriculture, the environment, and human health.
Good day, everyone. My name is Angelica Santos from BPL Student. I will discuss about introduction of GMO and production of GMO. First, what is GMO? GMO, or genetically modified organisms, are created by altering the DNA of a plant, animal, or microbe using modern biotechnology methods. This modification can involve the addition, deletion, or modification of specific genes to achieve desired traits or characteristics, such as making crops more resistant to pests or diseases, enhancing their nutritional value, or improving their shelf life. The purpose of GMOs is to address various agricultural and food production challenges, ultimately aiming to improve crop productivity, reduce the use of chemical pesticides, and provide benefits for both farmers and consumers. This is the example of GMO. Corn and soy are two of the most commonly grown GMO crops in the United States. The first one is corn. A significant portion of corn ground in the United States is genetically modified. These GMO corn varieties are engineered for traits like resistance to certain pests or herbicides. This modification helps improve crop yields and reduce the need for chemical pesticides, benefiting both farmers and the food industry. The second one is soy. GMO soybeans are primarily used for various purposes. One common application is in animal feed, particularly for poultry and livestock. With genetic modification, the soybeans can enhance their nutritional content and make them more suitable as a feed source for livestock. Additionally, GMO soybeans are also used for producing soybean oil, which is widely used in various food products. The use of GMO crops like corn and soy has become integral to modern agriculture and food production, contributing to increased efficiency and productivity in the agricultural sector.
Hello everyone, my name is Jonathan Vergara and I will discuss history of GM. The 1970s, the early beginnings of genetically modified organisms can be traced back to the 1970s when scientists started experimenting with genetics engineering techniques to modify the DNA of organisms. This period was marked by significant advancement in the biotechnology and emergence of recombinant DNA technology. One of the key milestones in the early 1970s was development of recombinant DNA technology by scientists Stanley Cohen and Herbert Boyer. This breakthrough allowed scientists to cut and combine DNA fragments from different sources, enabling them to transfer genes between organisms. In the 1980s, Stanley Cohen and Herbert Bayer created the first genetically modified organism, GMO. Using recombinant DNA technology, they inserted a gene from a toad into Escherichia coli's DNA, allowing it to produce galactosides. This breakthrough opened up new possibilities for genetic manipulation and paved the way of advancement into biotechnology and genetic engineering. Since then, scientists have a development and variety of GMOs used into agriculture, medicine, and environment research. In the 1990s, the commercialization of genetically modified organisms GMOs began with the introduction of the flour preserved tomato in 1984, other GMO crops such as soybeans, corns, and cotton gained commercial success in the 1996 despite controversy surrounding safety, environment impact, and labeling requirements. The commercialization of GMOs continued to grow. Today, GMO crops are widely cultivated and consumed in many countries, with application in industrial medical research. And disease control critics and raise concern about potential health risks and environment effects. In the 2000, the field of genetic modified organism GMOs experienced significant growth due to scientific advancement, agricultural application, increased adoption, and growing need for biopharmaceuticals and industrial compounds. New techniques like CRISPR, Cas9 enable more target gene editing. GMO crops like insect-resistant Bt cotton, herbicide-tolerant soybeans, and golden rice were development with increased adoption in North and South Korea, America. However, concern about environment impacts, human health, and corporation control over agricultural resources sparked debates. Despite this challenge, GMOs continue to be used in various industries, including pharmaceuticals and industrial compounds. In the 2008 brought new advancement in genetic editing techniques, expanded commercial use, ongoing debates surrounding GMOs. The discourse focus on genes rights, labeling, regulating public perception, and international trade disputes. The complexities of GMOs continue to raise important questions about agriculture and environment, sustainability, consumer choice, and the role of scientific research and policy making. In 2020, there have been significant developments in the field of genetically modified organisms. GMOs, these advancements have primary focus on various aspects including agriculture, medicine, and environment sustainability. Here's an explanation of some ongoing development in GMOs in 2020. Agriculture. Genetically modified crops have gained increased attention in 2020. Scientists are developing GMOs that are more resistant to pests, disease, and extreme weather conditions, aiming to enhance crop yields and ensure food security. Additional efforts and being made development generally modified crops in with approved nutritional contents. Gene editing Gene editing, the utilization of gene editing tools such as CRISPR Cas9 has been major focus on GMO research. These tools allows precise modification to an organism DNA, enabling scientists to edit specific genes responsible for disease traits. In 2020, this technology has been used to develop disease-resistant plants, improve crop productivity, and produce healthier livestock.
Creating a GMO plant involves a specific process. The first process, trait selection. Scientists start by identifying the desired trait they want to introduce into a plant, like resistance to drought, herbicides, or insects. This trait could be found in nature within the genes of various organisms. The second process, gene identification. Researchers locate the specific gene responsible for the desired trait in an organism that naturally possesses it. The third one is genetic modification. Using advanced biotechnology techniques, scientists extract the identified gene and insert it into the genome of the target plant. This process is often done with precision, ensuring that the new gene integrates seamlessly into the plant's DNA. The last one process is trait expressions. Once the gene is successfully integrated, the modified plant begins expressing the desired trait such as drought resistance or pest resistance. The purpose of this genetic modification is to enhance the characteristics of the plant to meet specific agricultural or environmental challenges, ultimately benefiting food production, sustainability, and resource conservation.
Good day everyone, I am Elisha Susana and I am here to discuss the step-by-step process of GMO. The first is identify. They start by identifying the specific trait they want to introduce into a plant. This trait could be something like resistance to insects, herbicides, or drought. Once they have identified the desired trait, they then search for a gene source that already possesses this trait. This gene source can be found in various organisms such as plants, animals, or microorganisms. For example, when scientists wanted to create insect-resistant corn, they searched for a gene source that could provide this trait. They found a gene in the soil bacterium Bacillus thuringiensis, or BT, that produces a natural insecticide. By introducing this gene into corn plants, they were able to create genetically modified corn that is resistant to insects. After identifying the gene with the desired trait, the next step in making a genetically modified organism is to make a copy of that gene. This process is often referred to as gene cloning. In the case of BT corn, scientists duplicated the gene responsible for insect resistance from the BT bacterium. They isolated the specific gene from the bacterium and then made multiple copies of it through a process called DNA cloning. This involves inserting gene into a vector such as plasmid, which can replicate the gene in host organism like bacteria. By duplicating the gene, scientists can generate a sufficient amount of the desired trait to be introduced in the target organism. In this case, corn plants. This step ensures that there is enough of the gene available for further genetic modification. After duplicating the gene with the desired trait, the next step in making a genetically modified organism is to insert this gene into the DNA of the target plant. This process is known as genetic transformation. In the case of BT corn, scientists insert the BT gene into the corn plant's DNA. This is done by using specialized tools and techniques such as genetic engineering or biolistic methods. The BT gene is carefully integrated into the DNA of the corn plant, allowing it to be expressed and produce the desired insect resistance trait. It's important to note that the newly inserted gene does not alter the plant's other existing traits. The goal is to introduce a specific trait, such as insect resistance, without affecting other characteristics of the plant. By successfully inserting the gene into the plant's DNA, scientists can create genetically modified crops that possesses the desired trait. In this case, insect resistance in corn. After the genetic transformation of the plant, the next step in making a genetically modified organism is to grow and test the newly modified plant. This step involves a series of controlled experiments and evaluations to ensure the safety and effectiveness of the GMO. In the case of BT corn, scientists first grow the modified corn plants in controlled environments such as greenhouses. This allows them to closely monitor the growth and the development of the plants, as well as observe the expression of the introduced trait, which in this case is insect resistance. Once the plants have been successfully grown in the greenhouse, scientists move on to conducting small-scale field tests. These field tests are carried out under controlled conditions to assess the performance of the GMO plants in a more realistic agricultural setting. They evaluate factors such as growth, yield, and the effectiveness of the introduced trait. Throughout this process, thorough reviews and evaluations are conducted to ensure the safety and regulatory compliance of the GMO plants. These reviews involve rigorous assessments of the potential environmental and health impacts of the modified crops. The entire process of testing and evaluation can take several years to complete. Only after the GMO plants have undergone extensive testing and received regulatory approval can farmers begin using them in their agricultural practices. The development of GMO is a complex and carefully regulated process that aims to harness the power of genetic engineering for the advancement of agriculture and the betterment of our society.